the next speaker, Luisa Fernanda Bermudez Salazar from the INTA CONICET from Argentina. She's going to talk about deciphering the epigenetic variability in Solano-Lycopersicum accessions and their influence on hybrid vigor. So you have 15 minutes, including questions. So the floor is yours, Luisa.
Okay, can you hear me here?
Yep.
Okay, thank you. Okay, good afternoon to everyone. Thanks for being here, and thanks to the organizer to give me the opportunity to present my work that I have been doing during this last year. So I'm so excited to be here. So let's talk about the deciphering the epigenetic variability in the Solano-Lycopersicum and its influence on hybrid vigor. So just to start as a background, as we know, the heterosis or hybrid vigor is an extensively used tool for animal and also plant breeding, and it's mainly used for improving growth, yield, and some other quantitative traits. Even when this phenomenon has been studied over a century, there are not complete consensus about the mechanisms that are involved. Right? Several models have been proposed to explain the heterosis phenomenon as dominance, overdominance, and also pseudo-overdominance. But recently, even when genomic scale studies have reinforced the participation of these models, also show that there are a genome reprogramming and there are some interaction between divergent genomes. So having this in mind, recently some authors proposed a model where the differences observed between hybrids and the hybrids are because of the most efficiency in the use of the energy. So principally is related with low energy used in the recycling of proteins and RNAs and used in the growth or cell division. In this case, what they propose is that hybrids show a higher metabolic efficiency based on the choice of the alleles to be expressed. And this selection is based on the stability and efficiency on the encoded protein. So in the inbred lines, they cannot choose since they are homozygous and they degrade more proteins produced from weak alleles. And in the hybrids, they choose and downregulate these weak alleles. Right? So this downregulation is thing to do by a global gene express shift via epigenetic regulation, especially cytosine methylation via RNA-directed DNA methylation. So having this background, what we proposed is using a plant material that are 6 tomato lines used that were previously obtained and genomically characterized by Dr. Mathilde Kau's group in INRA Avignon. So these lines represent a really large range of genotypic diversity, comprising about 96% of the species diversity, and are 3 lines from large fruit, Levoville, LA147, Ferrum, and 3 cherry fruits, Plovdiv, Serville, and LA1420. These 6 lines, as you can see here, present a large phenotypical diversity, and some of them are breeding lines and some of them are land races from Ecuador and Honduras. So with these accessions, they performed some crosses, Levoville and Serville, Ferrum and LA1420, and LA114 to Plovdiv genotypes. These are the crosses that we used. So with this material, we performed yield and quality tracing field condition in 2 years to identify agronomical transgressive traits. Also, we performed and analyzed an RNA profile, small RNA and methylome profiles in duplicate from green fruits in cell expansion stage. And we performed an integrative analysis of the aim to identify genes or metabolic pathways regulated by epigenetic mechanisms that could be underneath heterosis. So as a result, the first thing is that we perform a study to identify the transcriptional and epigenetic diversity in the parental lines, right? So the first thing that we found is that even when whole transcriptional profiles of these parental lines showed a high similarity between all the genotypes, after statistical analysis, we found over 8,000 genes with differential expression in at least one of the parental genotypes, right? The differentially expressed genes were grouped into three main clusters depending on their expression levels. And it's important to show that the most distant cherry fruits, Plovdiv and Serville, group together in this pattern of the transcriptional profile. So in line with the aim of deciphering the epigenetic mechanism regulating the observed diversity at the gene expression levels, we further investigated the small RNA accumulation clusters in those genes, right? We found here over 4,000 differential accumulated genes, even on regulatory region or gene body. And again, we found three main clusters that we observed in this accumulation. And again, Plovdiv and Serville genotypes, the cherry fruits group together. Later, regarding the methylome profile, we found over 144,000 differentially methylated regions along the whole genome, and the hierarchical clustering of the most differentially methylated region, that is what we are looking in here, grouped the cherry fruits accession, all the three of them, including the landrace LA1420, and the DMRs were arranged into two main clusters. The first cluster, each genotype displayed, as you can see here, a clear unique group of hypermethylated regions, as you can see in here for all the genotypes, and the second cluster show a hypermethylated region exclusively for the LA147 genotype, right? So having in mind which is the genome-wide association between these three data sets, what we can see is that we found that the differences in the methylation in all the parental comparisons we performed in the three parental comparisons is that they are strongly associated with the accumulation of a small RNA. So when you can see here the genes that show down hypomethylation of the regulatory region are associated significantly with the small RNA accumulation for the three comparison, but no association was found for the transcriptome profile. Yes, and this was true for the up methylation when it was hypermethylated in the cherry fruit, right? So you can see here the genes that show hypermethylation on the promoters that are strongly associated with a higher accumulation of a small RNA cluster, but no association was found with the transcriptome profile. Even though we were looking for which genes that show differential methylation levels in the regulatory region and also a small RNA accumulation in the regulatory region different, that relating to the transcriptome profile different. So we found around 700 genes that showed the three difference in these data sets. And when looking at these 700 genes, we found that most of them are associated and are enriched into the some metabolic processes and also in the transport categories. Just to show you an interesting example is the VASHL gene, when we found that there are a really nice negative correlation between the transcript profile and the small RNA profile over the promoter region. More interestingly is that we found that this accumulation of small RNA in the promoter region can be also associated with higher methylation levels on this promoter region. Yes. So, okay, regarding the hybrids characterization, as I mentioned, the parental one is those lines with large fruit. Just parental two is the cherry fruit, just to memorize that. Okay, so what we perform is yield and quality traits in experiments in field condition. We perform in Mendoza, in Argentina, when we have the highest direct solar irradiation of the globe in the collaboration with Dr. Iris Peralta. So what we found is that we found some transgressive phenotypic traits on field trials, principally related to plant weight, number of branches, and number of inflorescence. For some hybrids, we found some transgressive traits in fruit's total weight. And we performed, as I mentioned, in 2 different years. And for plant weight and number of inflorescence, these transgressive traits were consistent in both years. So regarding the genes at the differential expression and small RNA cluster accumulation in the hybrids, we found that the levovil and servil was the one with the highest number of loci with differential accumulation of a small RNA cluster. And it's important to note that these are the most distant accessions that were crossed. And on the other side, LA147 Plovdiv hybrid show the highest number of differentially expressed genes compared to their parentals, and the other hybrid Ferrum plus 1420 is like in the middle of both other hybrids. So just to remember that as we can calculate the trait inheritance, we can see that we can have additive inheritance in the middle of the two parentals, some dominant and also recessive, and we have other like transgressive characters that can be over dominant or over recessive. So what we look until today is that we have 25% of the traits evaluated like in a transgressive inheritance. And when looking about the inheritance patterns, the inheritance pattern of the transcript level and small RNA accumulation, we found that the levovil and servil hybrid showed the higher percentage of genes with that were non-additively inherited for the differential expression, and also with the small RNA accumulation, principally when you are looking in the comparison to the cherry fruit. Regarding the Ferrum and LA1420, we found some similar pattern with a nice number of genes with a transgressive inheritance regarding the small RNA accumulation and also the transcript level. And regarding the methylation level and methylation profile in those hybrids, we found like 140,000 DMRs and 130,000 DMRs in these two hybrids, levovil and servil and Ferrum LA1420. And what we found is that there are many DMRs that show the high percentage of non-additive inheritance. As you can see here in green is the additive inheritance, but there are many that are dominant, recessive, and also transgressive inheritance, right, in these hybrids. So when looking for those differential expressions in the hybrids and which of them presents DMRs on their regulatory region or also a small RNA differentially accumulated in the regulatory region and also for gene body, we found like a nice percent that it's associated with also the differences in methylation and small RNA accumulation. Even when there are some of these clusters of small RNA that are not differentially by statistical, we went to check which of those differentially expressed genes that present the differentials in methylation and presence of a small RNA cluster that can be putatively regulated by RNA-directed DNA methylation. We saw that the binding category and the catalytic activity functional category was really higher in these hybrids, and it can be remembered some of the or initial hypotheses that the recycling of the proteins and mRNAs can be regulated in these hybrids. So just some remarks that the differential expressed genes that are epigenetically—
You have 2 minutes.
Okay, that are epigenetically regulated in the parentals can be associated to key metabolic pathways, that the epigenetically mechanisms seem to be involved in the regulation of the gene expression in the hybrids, and the hybrids show genes related to protein and mRNA metabolism and catabolism that can be regulated by RDDM mechanism. And a vast reservoir of data we generated, and I hope that will be released soon, bringing a new level of understanding about how the epigenetic mechanism act on hybrid vigor. Next steps is we want to integrate all these data sets obtained here with those already published by Dr. Matthew Kau's group with metabolome, proteome, enzymatic activities, and organoleptical quality. These, together with the agronomical trait that we already tested, it will be nice to check into integrative approach. So thanks to the group, when I did all this work, the Dr. Fernando Carrari's group and my colleagues, and Dr. Magdalena Rossi group in University of São Paulo in Brazil, And also Nicolas Velora, that is the bioinformatician that really helped me with all of this data set. Dr. Iris Peralta in Mendoza, and Dr. Matilde Kaus in INRA, and Dr. Giovannoni, Dr. Fee when I performed the methylome assays. So thanks for attention, and I will be happy to take some questions if you have.
Okay, thank you very much. Nice presentation. So we have 2 questions that I can see. One of them is for Leandro Quadrana. He said, very nice talk, Luisa. I wonder if you look at the association between variation in DNA methylation and transposon expression. In other words, can someone of the hybrids show transposon reactivation as proposed by the genomic shock hypothesis?
Yes, we are working on that. Actually, the first thing that we wanted to perform is like to check the carefully annotation of those transposable elements. But like this is a huge work that we have done, and we are working on that because it is really associated with some specific methylation in some of these transposable elements. So we are working on that, but I didn't have time to show to you in this 10 minutes.
Okay. We have another question that I think that is similar. Let's say, hello, Luisa, great talk. Since RDRDM is involved, did you check how many of the DMRs and loci with differential sRNA enrichment are associated with transposable elements? I think that in some ways you answered, but you want to add something else?
Yes, actually is another part of the work here. I choose to focus on the differential express genes, but for sure that the transposable elements, like it's a huge part of the work that we are exploring too. But yes, and it's really associated, the small RNA, even when we are, I mean, when we plot all the mapping of the small RNAs over the genome, what we find is that there are like really strong association with the small RNA cluster in the genic region. That we know that in tomato genome, we have like the extremes of the chromosomes with enrichment of genes. And we can see that the small RNA clusters are really mapping on that, not in the middle of the centromere where we have the transposable elements. But even though what we are looking for is that some of those transposable elements on the promoter regions are really associated with the main methylation differences. So it's like we can divide some of the transposable elements associated with genes and some of the centromeric region, but it's a really a lot of things that we have already to do.
I think that we will have more time for discussion in the break. Okay, so Jose, you want to introduce our next speaker?
Yes, so the next speaker is Lara Pereira. She's a postdoc in the University of Georgia, and she's going to speak about natural genetic diversity in tomato flavor genes. So you're there? Yes. So can you share your screen, please?
Sure. Can you see the slide? Hello?
Yes, the slide.
Yes. Is there?
Okay, so good afternoon. I'm gonna talk about tomato flavor. So the first thing I want to do is just introduct a brief introduction about flavor. Flavor is determined by a combination of sugars, organic acids, and a huge array of volatiles that are synthesized during fruit ripening. Tomato has lost flavor during crop improvement, mainly because the breeding process has focused on agronomic traits as yield and disease resistance. But a huge effort has been done recently to try to identify the metabolites that are more important for flavor. So for example, here in the left, you have an association analysis between volatile profile and liking scores from a panel of tasters of different accessions. And with this analysis, they identified several metabolites that are really important for flavor, are positively associated as sugars, for example, and others that are negative as guaiacol or methyl salicylate that confer a smoky flavor in the fruit. And it's also important to highlight that volatiles are active in general at very low concentrations. So very small changes in the quantity of these volatiles in fruits can hugely alter the fruit flavor. Flavor has also changed during crop domestication, as it is part of the domestication syndrome in fruit crops. So we were interested on analyzing the genetic diversity of flavor genes in the baryton collection. The baryton collection is comprised by diverse germ plants from South and Central America and has been recently used to elucidate the domestication history of tomato. The closest wild ancestor of tomato is Solanum pimpinellifolium, that is found mainly in Ecuador and Peru. And it gave rise in the same region to SLC, Solanum lycopersicum cerasiformi, that is a semi-domesticated species that later migrated to Central America and gave rise to the cultivated tomato Solanum lycopersicum in Mexico. So we hypothesized that beneficial alleles or beneficial haplotypes may have been left behind during this whole process of domestication and breeding and could potentially be recovered to improve flavor. We have already studied one known flavor gene using the baryton collection and several other resources. This gene is the non-smoky glycosyl transferase that is an enzyme that conjugates guaiacyl and methylsalicylate, negative volatiles for flavor, with trisaccharides and makes them non-convertible to free volatiles. So the volatiles cannot be emitted during consumption and they are non-smoky. And these genes are located in a region that was not well assembled in the reference genome. And we used long-read high-quality assemblies obtained and published in this paper to resolve the non-smoky glycosyl transferase haplotypes. So here, some of the accessions are from the baryton collection, and you can see how diverse this is, specifically in terms of structural variation. For example, we have some duplications, copy number variation in some of these genes, in one of these genes. And we also have a 23 kb deletion that completely deleted all the functional alleles of these non-smoky glycosyl transferases. And this deletion was associated to the phenotype, to metabolite emission in both the baryton collection and an F2 population developed from two Solanum lycopersicum cerasiformi SLC accessions from the baryton collection. So after this work, we thought that it would be very interesting to see if other flavor genes were also associated with metabolite levels in the baryton collection. And to check that, we used association mapping, and we found association between sugars, acids, and several volatiles to 5 known genes. Here, I'm just focusing on 2 of them. One of them is LIM5. This is a cell wall invertase that hydrolyzes sucrose into glucose and fructose. And it was highly associated to soluble solid content, that is mainly sugar measurement, in the baryton collection. And the other gene is the lipoxygenase LOXC that transforms linolenic acid into C5 and C6 lipid-derived volatiles. And these 2 genes and most of the other ones have been cloned using mainly interspecific variation with the distantly related green-fruited species Solanum pennellii. So we wanted to explore the genetic diversity in these loci in the red fruited clade and using the baryton collection. So we use the variants within the gene as well as 3 kb upstream and 1 kb downstream the gene. And the genetic diversity is presented here in this heat map. Each row corresponds to an accession and each column corresponds to a variant. And I also added some annotation of this information. So for example, here we have the location of the variant. This white region, for example, is the intron. And we have also annotation about the accessions, specifically the sugar content in this case, and the phylogenetic group. And well, the accessions clustered in 6 different groups. 3 of them were formed mainly by the SP and SLC from Ecuador accessions, and they were highly divergent compared to the reference. And 3 other groups were more similar to the reference and contained fewer alternate alleles. And I want to highlight specifically group 5, cluster 5 and cluster 6. Cluster 5 was presented the lowest amount of sugars and was identical to the reference genome. And the cluster 6 presented only 3 conserved variants in the group and presented the highest amount of sugars. So the 2 of these variants here highlighted with the black rectangles were associated to the phenotype in the baryton collection. Next, we constructed haplotype networks. Each circle corresponds to a haplotype, and the size of the circle is proportional to the number of accessions carrying that haplotype. In the left, the haplotype is colored by the phylogenetic group, and here you can see how we have in total 24 different haplotypes, and most of them are unique. So they are carried only by 1 or very few accessions. Specifically, the Peruvian SP that are very diverse have very unique haplotypes. One of them is probably the most likely ancestor of tomato gene. And well, we can also see these 2 more common haplotypes were found in the Central American accessions mainly. And the most common haplotype was the one we found in the Lycopersicum group and a different group of SLC with different origins. In the right, we have colored the haplotype network using the amount of sugars in this case. And we found that it's remarkable the difference between the most common haplotype and the second most common haplotype. Only 1 mutation is found here between these 2. These lines corresponds to mutations, but in the most common haplotype, the sugar content is low. And in the second most common haplotype, the sugar content is intermediate to high. So we hypothesize that it's very likely that that mutation that actually provokes and causes an amino acid change can impact protein activity. To try to check that, we first did a modeling of the protein. The residue is marked here in red. And we observed that actually it was predicted to have low impact in protein structure, but this SNP was, again, highly associated with the phenotype. So our collaborators performed enzymatic assays and found that actually the enzyme, when it carried the alternate allele, was more active than the reference allele. So we can say that this amino acid change actually is causative for the phenotype.
Lara, you have 2 minutes left.
The other example is in LOXC, where we found a lot of heterozygosity. So we hypothesized that this heterozygosity, since it's not very common in tomato, could be due to an ancient duplication that was absent in the reference genome. And to check that, we used again these long read assemblies, specifically one pimpinellifolium accession from Peru. And we found actually that it was the gene was duplicated. So in the Peruvian accession, we found a reference copy and a second copy duplicated in tandem that was associated with the volatile content. We also align Illumina reads to this new assembly and found that in addition to the duplication, we also observed other several structural variants in other accessions, indicating that, as we already saw in the non-smoky glycosyltransferase, these flavor genes are usually affected by structural variation. To end my presentation, I just want to highlight that in the baryton collection, we found high genetic diversity. In most cases, the mutations that we observe are different from the described causative mutations. And we think that these genes have a role in tomato domestication because they overlap with selective sweeps. And finally, the objective is to find haplotypes that could be beneficial and are absent within the cultivated tomato. We are seeing that this is the case, and some of these haplotypes can be introduced into breeding pools and ease the breeding process to improve flavor in tomato. And just to end up, I want to thank all of you for listening, and my research group, specifically Esther Vandernaam, my supervisor, and Manoj, that has performed a lot of the bioinformatic work, and all our collaborators.
Thank you very much, Lara. This was a great presentation. So there are no questions coming. Maybe we should leave a couple of minutes, but I have plenty of questions. So one of them is so you find these alleles of LIM5 and LOX-C, no? I think it's the name. And so these are natural alleles, but are they always between cultivated and Seraciformes? So are they already used in cultivated tomato?
We are exploring that, but in our preliminary results, most of the haplotypes, the thing is, we are seeing a lot of haplotypes, but most of them are absent in cultivated tomato.
So usually when you find, because you have phenotypic data for flavor, for sugars, etc. So usually when you find, because at the level of sequence, we know that Seraciforme and Pimpinellifolium have much more sequence diversity than cultivated tomato. But at the level of phenotype, is this true also? You find much more diversity? So it's always in this direction.
It's not always the case. In some cases, for example, in this group, in cluster 5 for LOX-C, the accessions are very similar to the sequence is very similar to the reference. So they are very similar to cultivated tomato. And here we have a lot of Seraciforme, but we observe a huge diversity in phenotypes. As you can see in the violin plot here for cluster 5, the levels of the volatiles present a huge range. So in cases like this, we are considering that one of the possibilities is that there are other genes segregating in this genetic pool that are not identified yet. So we are not taking these genes into account, but they are also important for the accumulation of this volatile.
Okay, we have now questions coming in. So David Cano is asking you, do you know which wild cultivars the haplotype LIM5 associated with higher enzymatic activity comes from?
I don't know which cultivars specifically, but in the case of the allele of LIM5, is one of the cases that is actually, this amino acid change is actually observed in some cultivated accessions. So that specific allele has been already integrated into breeding pools.
Okay, and then we have a last question. So did you, well, thanks for your presentation. Did you investigate the relationship between LOX-C allele and apocarotenoid box accumulation? This is Mateo Martina.
Yes, we have observed we have performed the local mapping association also for apocarotenoids, and some of them are significantly associated. As it was already reported I'm sure that's why the person is asking.
He's referencing the paper, the Gao paper.
Yeah, we found the same.
Okay, thank you very much. So we go to the next speaker.
You.
Okay. Thank you. So now we will switch to Gustavo Silva Arias from the Technical University of Munich, the TUM. And he's going to talk us about the ecological genomics in wild tomatoes to reveal mechanisms of adaptation to living in the driest region of the world. So I think that the floor is yours, Gustavo. I think that I cannot see you.
Hello.
And I can, yeah.
So I will share my screen now.
Okay, yeah.
Good. Yeah, you can hear me well?
We can hear you and we can see you and we are seeing your screen now. So the floor is yours. Again, you have 15 minutes, including questions.
Okay, thank you.
Yeah.
So thank you, Aureliano and Jose, for having me and for the organization of this nice meeting. Today, I will talk about ecological genomics on wild tomato. I will focus specifically in Solanaceae chilense. So this species is very interesting because it inhabits very harsh environments. It occurs between South Peru and North Chile, and we can see a lot of morphological variation and climatic variation between this range. The species can be divided in 4 main genetic groups, these central groups, and 2 main southern groups. There is one in the coastal and other restricted to the highlands. There is a 4th group towards the north that could be more divergent, but we still have not much information on that. So we are more focused in the divergence of these 2 southern groups. We already did some whole genome and some estimations of the historical demography of these 2 groups, and we can find that the coastal group diverges first than the highland group, and we can see here in the estimation of the population sizes in the central group, the population size are quite constant through time, while in the 2 divergent groups it diminishes towards the recent times and is stronger in the coastal populations there. So we can assume that these are more isolated populations and diverse first, so they have a stronger drift and have less variation. If we put in context this with the other wild tomatoes, we can see that Solanaceae chilense occurs in the southern extreme of the distribution of the whole group, and also it occupies the driest and coldest environmental positions there compared to the other wild tomatoes. I also see like some update of this information about the environmental perspective of the wild tomatoes in one of the posters published with Gabriela Ramirez Ojeda. So if we focus on Solanaceae chilense, we can also see a strong variation in the environmental distribution between the different populations, being the coastal and the highland populations quite well differentiated from other populations from the environmental distribution. And if we see some specific climatic variables, we can see, for example, the highland populations tend to be distributed in regions that have more or strong variation during the day and during the year in the temperature that is typical in highland and very dry environments, and also occupied environments with a stronger potential level of transpiration. While the coastal is a little bit different, it tends to occupy higher temperatures, both for the coldest and warmest streams, but also it seems that it occupies a wider environmental space in these cloud coverage variables there. So in the coastal, we can see a strong effect of the moist, especially in the mornings, and it seems to be very important in the distribution of the plants and how they adapt to this environment. So taking into account that these 2 divergent groups occupies very differentiated environments, we want to understand how is the gene network evolution to be able to colonize these different environments. So the first attempt was done by Katarina Bondel using 16 abiotic stress response, and they consistently showed that there are different diversity or variation patterns of the genes depending of the function in the networks. For example, the functional genes have more non-synonymous variation than synonymous variation, and the regulatory genes have the opposite trend. Also, there are some specific genes that have to be more homogeneous selection across the whole species range. Well, there are some specific genes that could be more specific, like selection signatures in some of the populations, showing that there are a local adaptation. We also trying to attempt this approach, studying some pathogen-resistant genes, and we found some interesting pattern there, that some hub genes or central genes in the networks seems to be more differentiated between different regions. Means that you need like strong changes to really colonize new environments, while some peripheral genes have more differentiation within the regions, showing that you need like some kind of fine tuning with some specific genes to be able to colonize some specific environments within a whole region. So you can hear more about this from the experts. Yesterday we had some nice talk in the diversity and evolution session, and tomorrow in the stress biology, you can also find some details of this, of the evolution of these genes network, pathogen-resistant genes. So how we are trying to address this gene network evolution in Solanum chilense. First, we are trying to use population genomics approaches using whole genome sequence. So basically, we sequence 30 individuals across the whole species distribution in 6 different populations. So first, we study the genetic structure, and it shows like these 2 coastal populations, for example, that you can see here, are strongly differentiated from the others. And also, if you study more in detail the differentiation between the central and highland populations, you can see a more finer differentiation of these highland populations. With Kai, we are trying to study which possible genes could be involved in the adaptation of these divergent populations. So we run different methods to detect positive selection as a hard sweeps with different methods in each of the populations. Our first main result is that there are difference in the amount of genes involved in positive or in local adaptation between different regions or populations. Being, for example, in the highland populations, there are more positive selected genes, or at least that showing this signal, while, for example, in the coastal, we can detect less amount of genes. Also, we see that there are strong differences between the populations in the genes itself that we detected. So given this high or very big list of candidate genes on their positive selections, we are first the classic functional annotation approaches, and using this enrichment, we can like subset these different genes in genes that have a similar function that can be clustered in some gene networks. And for each of the subsets of the group of genes involved in similar or that have similar functional annotation, we applied a genotype environment association analysis to see which populations and which specific SNPs could be associated with some specific climatic variables. From that, we can associate, for example, in these specific genes that I'm highlighting here that could be more related with circadian rhythm and response to light. We can see a strong differentiation in the 2 highland populations, this like the highland in the south and one the highland in the central populations, and we can subset this specific set of polymorphisms that are present in these populations and understand better the polymorphisms. From that, we can, with this specific set of genes and polymorphisms, we can test some specific hypotheses. For example, if the genes comes are de novo mutations or mutations that are in the standing variation on the whole species range and become adaptive in that specific populations. We also are planning an alternative approach to detect this network evolution in the colonization processes using fine-scale landscape approaches. Basically, we do a zoom in the sampling in several populations. So in that case, in this specific case, we sample 18 populations in the whole range of the species, but focusing more in the central region because we want to detect this like a slightly process of the, let's say, for example, the climbing to the highlands or to going to the south to the driest regions. So we're sampling a different amount of populations in 4 different valleys, trying to touch this gradient, altitudinal gradient. And also we included some scattered individuals from the divergent populations. Today, I will show you some of the results in some target genes that we include in this sampling that are genes related with drought and cold response previously identified in gene expression analysis. So first, I can show you the general population structure detected in this data set. We can see here again this external differentiation of the coastal population in these green dots. And the main differentiation process pattern, you can see the geographical pattern. These brown points are the northernmost populations. These are more the 3 central valleys plus the highland south populations that are very well clustered with the other central populations and the coastal populations. To be able to see the differentiation of the highland subpopulations, we can study all variation accounted in this principal component 4. And so we decided that studying these 4 components will be enough to characterize the population structure. And from that, we detect some outlier losses that are basically variants that depart from these main differentiation patterns. So for each of the structure of the main principal components, we can identify different sets of genes that in total are around 600 out of 22.6 thousand genes. So the previous approach are based in the basic mapping and variant calling approaches, but in Solana Chile it's a little bit tricky to have like a good reliable set of.
You have 2 minutes.
Okay. To detect these variants because there is a lot of diversity, heterozygosity, and indels. So we also are trying to assess this variation by de novo assembly. So basically, we take all our target genes, the reads, and perform first de novo assembly without any reference, and then we study each of the contings to annotate to the different genes that we are targeting and detect, for example, some presence absence. That is the case that I am showing here in these plots. So these plots are showing the presence absence of different genes. So for the cold genes that we are targeting, 70 out of 233 genes show this presence absence variation, and you can see here in the x-axis all the populations in the latitudinal gradient. So our naive expectations here is to see some differences, for example, in the altitudinal. For example, here in the each of the populations labeled here, pay 1, 2, 3, or 4 are following this altitudinal gradient. we're expecting to see like some genes that will be more absent or present in the populations due to these differences in the altitude. But at the beginning, we can see that most of the genes are conserved, are present in over the whole species range. And then this is the general trend here, plus some genes that are almost absent in most of the places that could be more like accessory genes. From this set of the presence absent genes, only 11 showed some outlier variants there. For the drug genes, we more found a similar pattern there, that there are just like half of the genes also have this presence absence pattern, and just few have like outlier variants. So thank you very much for having me. I want to first acknowledge for all the team here in the population genetics, and also to Edgardo Ortiz, who is working with me in the de novo assembly of the study of the genes. So thank you very much.
Okay, thank you very much, Gustavo. I think that we don't have much time for questions, but if you answer this quickly, that would be great. So Marcos Gea Cortines asked you a question that is, are these changes in copy number are there changes in copy number in specific genes for certain clusters, such as circadian clocks of flower development in different Chilean accessions?
We are trying to study these copy numbers. We see differences in the insertion of deletions in the whole genome pattern, but for these specific genes, we still doesn't study specifically, and the most difficult part there is to really differentiate them, when we have, like, for example, when we de novo assemble some of the genes, it's very difficult to differentiate duplications from different alleles, because there is a lot of polymorphisms there. So this is one technical issue that we still have to deal, because we are using short reads. But for some, for example, of the NLRs, we are already able to detect some duplications of genes between different populations. But for the circadian genes, I have no results still.
Okay, thanks. We will continue the discussion later on during the breaks. Okay, so Jose, I pass the microphone or the turn to Jose and to introduce our last speaker.
Yes, so the last speaker is Salvador Torres Montilla. He comes from CRAG in Barcelona in Spain, and he's going to talk about a novel system for artificial differentiation of chromoplasts from pre-existing chloroplasts in leaves. I hope there. Yes, perfect. So if you can share your screen.
Okay, can you see it?
Yes, can you? Perfect, that's it.
Okay. Thank you, Jose. Thank you for the organizer for giving me this opportunity. Here I'm presenting a novel system for the artificial differentiation of chromoplasts from pre-existing chloroplasts in leaves. But first, let me introduce carotenoids briefly, that are 40-carbon isoprenoids that plants, as well as other organisms, are able to produce. They are pigments that give color to some fruits, some roots, and also some flowers, but also they carry out the other functions, like they participate in photosynthesis, collaborating with chlorophylls in light harvesting, and also they play different roles in photoprotection and signaling. Humans, we are not able to produce carotenoids, but they are essential for us, mainly because of their role as provitamin A. But back to plant, and in plants, carotenoids are synthesized in plastids, and they are present in chloroplasts in all green tissues. But also they are present in a different kind of plastid that are chromoplasts, that are not photosynthetically active, but they are instead specialized in the production and accumulation of carotenoids. In nature, chromoplasts are not developed in all plant tissues and not even in all plant species. But when they do, normally they are differentiated from pre-existing chloroplasts. This process of differentiation from chloroplast into chromoplast is not very well understood in nature, because when it occurs normally, like during the tomato fruit ripening, there are many other processes in parallel, like the ethylene production, the sugar accumulation, or the cell wall loosening. That makes really difficult to identify what are the molecules or the genes that are specifically responsible to trigger this conversion of chloro into chromoplast. Here, I'm presenting a novel system that is able to reproduce these mechanisms in leaves. And this mechanism is based in the expression of the CRTB gene that comes from a bacteria, from Pantoyana latis, and encodes for an enzyme that is able to catalyze the same reaction as the endogenous phytoin synthase, or PSY. This reaction is the condensation of two molecules of GGPP into one molecule of phytoin. This phytoin is the first molecule, the first carotenoid of the endogenous pathway, and is a molecule with no color. And in nature, it is not accumulated in leaves, not even when we overexpress the endogenous PSY. We are not able to see significant levels of phytoin accumulated, but when we express CRTB in Nicotiana ventamiana leaves, we are able to see a massive accumulation of phytoin and also increased levels of downstream carotenoids. But the most remarkable phenotype that we see is the change of color of the infiltrated areas from green to yellow, both when we compare it to the PSY and also when we compare it with GFP control. When we go more in detail into the structure of these infiltrated areas, we see that instead of observing the typical chloroplast that we are able to see in the control, we see that after the CRTB infiltration, the structure in plastid is completely modified. Actually, we see that thylakoid structure is completely disorganized, and this reminds us more to a chromoplast rather than to a chloroplast. To confirm if these plastids are really chromoplast, we wanted to compare our system to the natural one of the tomato fruit ripening. And for that, we use published data from a publication that they were measuring gene expression in different stages of the tomato fruit ripening. And we were focused on three, the mature green, orange, and red. But another possibility would be that instead of being a process of conversion of chloroplast into chromoplast, that this yellowing in leaves maybe can be a process of senescence. So instead of chromoplast, they can be gerontoplast. So to test also this hypothesis, we wanted to compare also with published data from a study that they were measuring gene expression in Arabidopsis leaves, comparing senescent leaves with non-senescent leaves. To be able to compare our system with the others, we also performed an RNA-seq analysis when we were using agro-infiltrated areas of Nicotiana ventamiana, agro-infiltrated with CRTB, 4 days after the agro-infiltration, when plastids are already transformed, and comparing them to GFP agro-infiltrated areas that were agro-infiltrated in the same leaves and also at the same time. We saw that the samples were clustering really well into different groups. And after the differential expression analysis, we saw that there was a massive change in gene expression with more than 5,000 differential expressed genes that were participating in more than 70 molecular functions and involving more than 100 biological processes. So what we did was to use these geoterms and to compare them to the other two systems. And then using those geoterms that were shared by the 3 different systems, we elaborated this heat map where we can see that the 3 first columns looks very similar, while the 4th one looks different. Actually, the first column corresponds to the comparison of our experiment in ventamiana, comparing CRTB against GFP agro-infiltrated areas. And the second and the third are the comparison in tomato, comparing the orange and the red stages to the mature green one. While the 4th one is the comparison in Arabidopsis, where they were comparing senescence. Something very similar we observe also when we take a look into the molecular functions, that the 3 first columns look similar and not to the 4th. So now we can establish that in our system, CRTB is able to trigger a chromoplast differentiation. And actually everything that describes this novel system is already published. And we can establish that it's similar to the natural process of the tomato fruit ripening. But as I said at the beginning, the advantage of our system is that it's more simple. We don't have the ethylene production, the sugar accumulation, so it would be easier to explore and to identify what genes and molecules are involved and responsible of that. Everything that I've been talking about by now is being focused on when plastids are already transformed. But now we want to pay attention also and be focused on what is happening before and in previous stages. thanks to our collaborators in the VTI in Cornell, we've been able to analyze a collection of different time points, also via RNA-seq. And this collection corresponds to different stages, where the first one is 1 day after the infiltration, when we already have high levels of high transcript levels of CRTB, but not yet high levels of phytoin accumulated. Then a few hours later, this phytoin started to increase the levels of accumulation, and then after that, after a few hours, more and more across the process. Also paying attention into the carotenoid levels, here is the ratio of the total carotenoid levels in CRTB compared to those one in GFP. We see that at the first stages, the ratio is around 1, so there are not increased their levels. But then after that, after a few hours, they appear to be increased. So at this point, we were expecting that probably at the very first stages, we won't see many or any differential expressed genes, and probably they will be appearing across the processes and increasing till the end. But this is not what we found, because at the very first stage, we already see that there are around 2,000 differential expressed genes, and in the second one, there are even more, around 2 times more, around 4,000. And then these genes, this number decreases in the third one, and finally increase till the end. So what we have here, attending to the numbers of genes, is that there are like 2 different events. Those, the first stages correspond to those changes that are before there's a big phytoin accumulation, and probably they are responding to the production of phytoin that is releasing some signal that is probably weakening the chloroplast identity and probably getting prepared to be transformed later. And then would be a second stage when these different time points will be already corresponding to the transformation of chloroplast into chromoplast, and that are those changes that are after the accumulation of phytoin and the increased levels of carotenoids. Now I am trying to get in more in detail into these genes, and I am under the supervision of Dr. Aureliano Bombarelli in Milano, trying to go further. And what I've been doing is just to compare these different lists between them and try to see how are the combination of genes across the different groups. And what we see here are ordered from the most abundant one to the less abundant. And what we can see is that the most abundant group of genes are those ones that are only present in the last time point. And actually, out of almost 6,000 differentially expressed genes that are present in the last time point, more than half of them, more than 3,000, are not present, are not differentially expressed in the others. So are only present in the last one. So are exclusively there. Then the second more abundant group of genes are those ones that exclusively appear in the previous time point. Then those ones that are only differentially expressed in the second, and then those in the fifth. And then after that, those are those genes that are present in 2, 3, 4, or more different set of time points. So the main feature here is that most of the genes are only differentially expressed in one of the time points. So these different time points are very different between them. Not only when we take a look into the genes, also when we take a look into which ontologies they are involved, we see something very similar. These are the biological processes that are upregulated, and actually we see that most of them are only present in one of the sets. The same with the biological processes that are downregulated, also molecular functions up and down. So the conclusion here is that we have a collection of different time points across the differentiation of chloroplast into chromoplast in Nicotiana bentamian allele, that we think that we can divide into 2 different phases. The first one would be before there are high levels of phytoin accumulated, and before that downstream carotenoids are increased, and would be a first phase that probably is related to awakening of chloroplast identity that is getting prepared to be converted after. And then a second phase that would be those genes that would be already related to the conversion of chloroplast into chromoplast. These are just preliminary results. I want to go further and be focused on that and extract more information. But the conclusions are that we have an artificial system that can develop the differentiation of chloroplast into chromoplast that CRTB is triggering, that this differentiation process could be divided in 2 different stages, those an early one and a more later one, And also that major changes in gene expression are specific of each time point. Also, the different time points are very different between them. So I want to thank all of you for your attention, to my supervisor and my colleagues here at CRAG, also our collaborators and the fundings. And if you have any question, I will be happy to answer.
Thank you very much, Salvador. That was very, very nice. So we have one question from Marcos Egea Cortines. He says he's asking if there is any effect of light in the process, or is it an activate and downhill process?
Actually, this is something that a colleague, another partner in the lab is doing his PhD, and he's more related in everything in light, and there's an effect. He's studying with far red and different lights, in white light and blue light, and so there's an influence, yeah.
And I was wondering, what's the difference between CRT and phytoenzyme on tomato?
Both the PSY, the phytoen, is the endogenous enzyme in plants, and CRTB is an analogous that comes from a bacteria, but is able to perform the same in Pantoia anatis, that is the bacteria, catalyze the same reaction. So it's producing phytoen from GGPP. So
these are homologs.
Yeah, the thing is that the sequence are really different, and also although it's able to go to the plastids, is both actually I have it here by itself. CRTB localized in cytosol and plastid, but also we have a version that only go to plastids, and when it remains in the cytosol, it's not able to trigger the phenotype, only the effects in plastid. By itself, the natural version is able, not only goes to the plastic.
Okay. So there are many questions. So are you planning to validate your RNA-seq data across the different time points by qRT-PCR, looking at specific gene families?
Yeah. We want now to find that in the prior RNA-seq, we are also trying to find some markers, and also we are very interested in try to have like what are the best genes to validate this RNA-seq. But for sure, this is one of the plans. Yeah. Okay. So
Joseph Hirschberg. What is the mechanism by which phytoen induces chromoplast differentiation?
Good question.
That's what you're trying to answer. You still have a way to answer.
Yeah, I would like to know. Let's see if I can answer in detail soon.
Lumi Shiose says, it's not a question, has a comment, says CRT catalyzes 2 steps.
It's like during all of this collection of time points, the transcript levels of CRTB remain similar, just at the very end, like 4 days after the infiltration, that corresponds to the last time point, the levels decrease. But it's like, I think that CRTB is just producing phytoen, and at the beginning, just this production, before it's accumulated, that probably is feeding the pathway, and probably, I don't know if something related to some downstream carotenoid is releasing some kind of signal. But then, because this doesn't stop, like it has to be like, if you interrupt the production of phytoen after that, the process is not able to advance. So you have to keep like CRTB working, producing the phytoen and feeding the whole downstream. And so both, I think that the accumulation of phytoen and also feeding downstream the pathway is what, like a combination of both events is what happens.
And one last and short question from Alan Gossent. Can you turn the process around, and would you see similar inverse gene expression profiles?
But how can I turn it around? Like how I don't know how to go from chromo to chloro, because actually when this kind of process after 4 days the expression of CRTB disappeared, but we've been studying like in long term, like after 10 days or 2 weeks, even 21 days. So it's not reversed. So I don't know how to go from chromo to chloro and to try to see if everything is happening the same, but in the reverse order.
Okay, thank you very much. So with this, Aureliano, I think we're done.
Yeah, we would like to thank all the speakers for their time. We want to also apologize, we ran out of time. We will be moving now to the breaking room, just in case that someone wants to have any discussions with the speakers. But also keep in mind that the poster session will start in almost 7 minutes, okay? So we still will move to the break session, and we can have some discussions there, but if you want to attend the posters, it will start in 7 minutes.
The link is in the chat.
The link is in the chat. You know, when we run out of the sessions in an online conference, it's not like in a live conference that you know that the coffee is outside waiting for you, and other sessions just starting to take all the cookies. So at least we don't have that. Okay, guys. So thank you very much. Thank you for your time. We will move now to the meeting room, and then you have questions, you can ask them there. Otherwise, we will see you tomorrow. Thank you very much.